It turns out that Canada's health agencies are still spreading misinformation about the novel modified RNA COVID-19 vaccines. And in today's report, I'm going to set the record straight by fact-checking their official parliamentary response. In response to Health Canada's unprecedented decision to abruptly withdraw the XBB 1.5 COVID-19 vaccines, order their destruction, and create gaps in vaccine availability, something the agency has worked diligently to avoid. Since the COVID shot rollout began in December 2020, Conservative MP Colin Carrie raised critical questions to the government regarding this move. In an order paper question called an OPQ, he asked if there were material differences between the XBB 1.5 vaccines and the new formulations that we see rolled out for 2024-2025, besides new mRNA coding for a different spike protein strain. And if so, what are they? He raises a crucial liability and legal question about vial contents, like how Canadians will be able to prove claims against the manufacturer if physical evidence is destroyed. Carrie inquires about how many vials will be destroyed and how they are broken down by manufacturer, that's Pfizer and Moderna, and how much this will cost the taxpayers. He probes the agency about a Global News article published in September that stated a provincial government official told them that the federal agency had started a new regulatory process for the vaccines, which resulted in a contractual obligation for Health Canada to withdraw all XBB products from the market until a lot could be released and distributed in Canada. Carrie asks what the nature of the contractual obligation is and who exactly is it with. He asks if Health Canada will see one of the residual DNA contaminants called the SV40 promoter removed from these new formulations, and if Health Canada will continue to assess these injections under the Centre for Vaccines, Clinical Trials and Biostatistics, or be transferred to another department. Now, before I continue, it's important to note here that the SV40 promoter is a genetic sequence derived from the controversial simian virus 40, SV40, that is commonly used in molecular biology and genetic engineering as a regulatory element in plasmids and other vectors to drive gene expression. It helps drive a process called transfection, acting as a switch that activates the production of a specific protein in cells. It's frequently used in laboratory settings to ensure high levels of gene expression in host cells, such as in the production of vaccines, therapeutics, and in research related to gene function. However, it has also been found to interact with the host's cell's genetic material, which can lead to unintended consequences like genetic mutations or cancer. These concerns are heightened when its presence had not been adequately disclosed or studied for safety in humans. Now, the Parliamentary Secretary to the Minister of Health, it's Liberal MP Yasir Naqvi, responded that when a COVID-19 vaccine is updated to target new virus strains, it is considered a modification of the existing product rather than a new drug. So a new drug identification number called a DIN is not issued. Instead, the previous vaccine versions are quarantined or removed from sale to prevent overlap. He says that this approach ensures that adverse events are correctly linked to the appropriate vaccine and aligns with practices followed by international regulators like the European Medicine Agency and the US FDA. But it seems like giving the same DIN to different formulations is actually a recipe waiting for confusion and disaster, and discrepancies around safety reporting, making it actually more difficult to track and differentiate the specific risks associated with each version of the vaccine. Naqvi states that the old formulations are no longer available because the updated KP.2 formulations have, quote, increased effectiveness, end quote. But before these new KP.2 injections were even authorized, provincial and territorial health authorities were reporting them as a strain that was dwindling in circulation. Public Health Ontario's weekly epidemiological summary on September 3rd said that KP.3 was the most prevalent lineage at 41.3%, and KP.2 was circulating in only 9% of sequenced cases. Public Health Ontario's latest summary on November 25th doesn't even include the KP.2 variant in its highlights at all. That's how irrelevant it is. Naqvi says that Health Canada cannot comment on the mRNA sequence of the Pfizer vaccine because it's confidential business information. But he did confirm that the SV40 promoter enhancer sequence was found to be a residual DNA fragment in the Pfizer-BioNTech COVID-19 vaccine, which I first reported on in April of 2023 with genomics expert Kevin McKernan. Naqvi goes on to repeat the public relations talking point that the fragment is inactive, has no functional role, and was measured to be consistently below the limit required by Health Canada and other international regulators. Except that in its own Clarifax documents, which are to seek clarification from the product sponsor, in this case Pfizer, Health Canada asked Pfizer for a whole host of clarifications surrounding the independent discovery of the SV40 promoter by Kevin McKernan, who previously told me that the agency's internal discussions don't match their public messaging.
The most interesting thing about those FOIA requests and those emails is that the communication they're giving to the public doesn't line up with the communication they have internally. So we've seen them go on record saying the DNA is of too little material or concentration to matter. Yet they're asking Pfizer for their protocols to measure this. So they asked for their qPCR protocols. So which is it? Is it too low to matter, or have you not measured it? It sounds like they haven't measured it. The other set of details in that email is they asked Pfizer to create a fragment length analysis assay, which Pfizer responded saying they don't currently have one and they're going to have to develop it and it's going to take some time. I think they gave them to December 1st of 2023 to sort that out. Well, that's very different than what they've communicated publicly, which is that these fragments are all under 200 bases. So how do they know that if Pfizer themselves doesn't even have an assay to assess that? What is this SV40 promoter and is it relevant? And Pfizer's communication back has been, it's not a material part of the plasmid for manufacturing. That's really disingenuous or an overt lie, because you can go through their EMA documents and see that they're in fact using kanamycin at, I think it was 50 micrograms per ml. So if they're using kanamycin, which makes complete sense that they would, the plasmid has a kanamycin selectable marker. So you can't get that plasmid to replicate in a coli without kanamycin present. And you need the promoter to do it. So without the SV40 promoter, they get no plasmid manufacturing. So they're trying to play this game, claiming it's immaterial, don't worry about it, don't look over here, when in fact, if you go through their EMA documents, you can see, and as anyone who understands how plasmids work, you can't get them to replicate in E. coli without the antibiotic resistance gene, and you need its promoter in order for that gene to be turned on. We have two things going on here. We have spike protein that is downregulating expression of P53, and we know we have SV40 promoters DNA in there that is also interacting with that pathway. This all needs to get untangled and better understood. The right thing would be to just get all that DNA out of there. But the second thing is you won't have a clean vaccine when that's done. If the spike protein does what it does, it's been reported to do, they need a different antigen, or they need to reconsider whether these respiratory viruses actually are addressable with injectable vaccines. The take-home lesson that I've read from reading those emails is that the regulators seem to be working hand in hand with Pfizer to help pave their excuse path, if you will, as opposed to asking them very adversarial questions over this contamination. It is clear from those emails that they were not informed of SV40 being in there. They discovered this upon our publication. They were given the sequence, but nobody took the 5 to 10 minutes it would take to pop that sequence into Snapgene and see the omission. So they had the sequence in hand, they filed it away and never looked at it until someone in the public showed them that, hey, there's something in there that no one consented to. And that particular sequence is all through the literature.
In addition to that, as also discussed by McKernan at the International Crisis Summit in Washington, D.C. this past February, the DNA inside of these injections are different than the DNA our regulatory bodies, including the World Health Organization, wrote these regulations for, making existing regulations outdated and inappropriately applied to these novel products. University professor Dr. Philip Buchholz discussed his similar concerns in a state Senate Health Committee, where he highlighted that previous vaccine regulations were inappropriately applied to these lipid nanoparticle-wrapped modified RNA injections.
Have a little bit in the vaccine. That's not a problem because naked DNA gets chewed up immediately upon vaccination, and there's no real mechanism for it to get inside the cells. They inappropriately applied that regulatory limit to this new kind of vaccine where everything is encapsulated in this lipid nanoparticle. It's basically packaged in a synthetic virus able to dump its contents into a cell. So I'm thinking Hanlon's razor here. Okay, I don't think there was anything nefarious here. I think it was just kind of a dumb oversight. And it's going to take, because the financial incentives are so great to just, sweep it under the rug, and the career incentives of people that approve this are going to be, there's nothing wrong here. It's going to take some encouragement to make people prove that it's okay. But I really believe this was an inappropriate application of an old school regulation.
Naqvi confirms that the modified RNA vaccines are reviewed in the Center for Vaccines, Clinical Trials, and Biostatistics of the BRDD. That's the Biologic and Radiopharmaceutical Drugs Directorate. Adding insult to injury, Naqvi says that the estimated number of COVID-19 vaccines in federal possession requiring destruction is approximately 670,000 doses of Pfizer, with an estimated cost to destroy of $137,000. Hardworking Canadians paid a premium to get these shots. They paid for the $10 million marketing ploy to subvert their subconscious into complying with vaccine uptake, and then they paid nearly $40 million for the vaccine injury support program meant to help those killed and maimed by these shots, but instead just lined the pockets of well-paid consultants, and now taxpayers pay to destroy the shots too. For Rebel News, I'm Tamara Ugolini. Every which way, taxpayers are footing the bill. It's time for a cleaning of house to restore trust and accountability and trim the bureaucracy. If you agree and want to send the leader of the opposition, Pierre Poilievre, a message to do just that, go to /thebureaucracy.com. That's /thebureaucracy.com.